Humans walk weird. Well, okay, a lot of animals walk weird, and fly weird, and swim weird. How about this? Humans walk weird for an ape. Apes are fabulously diverse in how we move, especially for such a small living family of animals. We have two different types of knuckle walkers, slow arboreal clamorers, high-speed brachiators, and of course, our bipedalism. My gentle and of course very modern apes. My name is Erica. This is my channel Gutsick, where I talk about human evolution and debunk pseudoscience. And today I want to tell you all about a new, pretty groundbreaking paper that explains how a major part of human bipedalism evolved. Now, of course, we're not the only apes that can or even do move bipedally, as you've no doubt seen on TikTok or Instagram or YouTube Shorts. It's just when other apes do it, they look kind of awkward. Despite having the same bones and muscles as humans, other great apes walk in a bent hip, bent knee style, and their knees kind of knock out to the side. As it turns out, this is a very inefficient means of bipedalism, meaning they burn way more energy than us when walking on those two legs. This is probably why they don't do it very often. I took myself outside to show you what this looks like. Humans will fully extend our hind limbs, often locking them when we're standing still. Chimps, on the other hand, when they're on 2 feet, are bent at both the hip and the knee, meaning they're essentially always in a partial squat. A consequence of this difference during bipedalism is that human bipedalism is smooth and energetically efficient, while chimp bipedalism is comparatively awkward and metabolically costly. Little apes like gibbons also move bipedally. In fact, unlike the great apes, these guys walk bipedally all the time when they're on the ground. Problem is, this doesn't amount to all that much strolling because they rarely come to the ground at all. The ability to walk upright, as well as the occasional use of that mode of moving, is called facultative bipedalism. Under that definition of facultative bipedalism, all apes are technically facultative bipeds, using bipedalism as a rare backup form of moving. If you're a nerd, you can think of this as their offhanded weapon. Humans, on the other hand, are obligate bipeds. This means we walk upright all the time when we're on the ground. We are obligated to do so. In this way, I wonder if you could technically call us facultative arboreal clamorers as well. Some people groups regularly climb to forage. Others climb competitively in a way that makes me think that they could at least keep up with a chimp group. Some of us can even do like 5 pull-ups on a good day and dead hang for a minute or more. You know, the really talented ones. Still, facultative versus obligate ways of moving reflect in our morphology or physical characteristics. Chimpanzees mostly knuckle walk on all 4 limbs, and so their skeletons and muscles reflect that. Humans typically walk upright on 2 legs, and so our skeletons and muscles reflect that in all its majestic weirdness. This is my human skeleton replica. Audio's gonna be a little echoey in here. I needed a blank wall, and my normal recording area has too many curios and knickknacks. Now, this guy was a biped like me and like you, presumably. And so what we're gonna do is we're gonna start superiorly or on top and move inferiorly and talk about all the characteristics that allowed him and us to be bipedal. Bipeds like us are upright. Our head sits on top of our vertebral column or our spine, and so because of that, the spine has to exit pretty far forward from the cranium. And so what that means is that the hole at the base of the skull, the foramen magnum, needs to be anterior or forward so that this spinal cord can exit directly below, maintaining this nice upright or orthograde posture. We can actually take a look at this by flipping the skull, or cranium in this case because the mandible is not attached, over and looking at where that big old foramen magnum actually is. You can take a pencil and stick it in there, and lo and behold, it's going to be nice and upright. We can compare this to something like a chimpanzee whose foramen magnum or hole at the base of the skull is more posterior or in the back. So we can take our pencil stick it into that foramen magnum, and what you're going to see is that it comes out at an angle as compared to upright, because these guys are knuckle walkers. It's more obvious when you look at them side by side with those palates held level to one another, and you can see that the human foramen magnum is indeed much more anterior than that of the chimpanzee. You got 5 types of vertebrae, singular vertebra, which I always mess that up, that make up your spine, and they're stacked on top of each other. The first type is your cervical vertebrae. You got 7 of those. That's actually super conserved for mammals. Then you have your 12 thoracic vertebrae that form your upper back, your 5 lumbar vertebrae that form your lower back, the single fused sacrum, which forms the posterior portion of the pelvic girdle, and then your little coccyx or your tailbone down here. And these vertebrae articulate with one another to give your spine an S shape. That shape helps you hold your body upright and anchors countless muscles that are critical for you to maintain your posture. Moving down to the pelvis, or the pelvic girdle, we see that the blades of the pelvis, or the ilia, are running front to back, or sagittally, to give your pelvis a bowl shape. It looks like you could hold either side of it and hold a bowl of soup in it. This allows you to anchor gluteal muscles in the back, which are, of course, critical not just for maintaining your stance, but for actually propelling yourself forward. This is really different from how chimpanzees have their ilia shaped and oriented, as well as their ischia shaped and oriented, but we'll get to that a little bit later. The femur or thigh bones are really long in humans to give us our long stride, and they come in at an angle to meet at the knee. We call this a valgus knee, because when you set this femur upright, you're going to see that it sort of juts off at an angle. This again allows us to carry our weight in a singular column. And then the last of our major characteristics that I expect you to know, God knows there's a lot of minutia here that I'm skipping over, has to do with the human foot. The human foot is unique amongst the apes in that the hallux, or big toe here, runs in line with all the rest of the toes. And this is important because when you step, you strike with your heel, you roll your weight through, and then you toe off with this big toe, and that propels you forward so you can catch yourself and then walk further, take another step. So that hallux runs in line with the rest of the toes. In addition, rolling that weight through requires 3 arches in the foot, or I should say is facilitated by 3 arches in the foot. So if you look at the bottom of the foot, what you'll notice is that it's kind of curved. You can feel your own foot, and check this out. I'm not going to show you with mine. There are some keywords banned in this comments section for a reason. In the human foot, we have 3 arches. You have an arch that runs on the medial side, you have an arch that runs on the lateral side, and you have an arch that runs transversely across the middle. So 2 longitudinal arches and your transverse arch. Those are going to be important for absorbing shock when you run, and like I said, facilitating that roll through of the weight. We also have a pretty big calcaneus or heel, but we could go on and on about the minutiae of the feet. All of this helps us to walk on 2 legs, a weird way of getting around when you think about it. We basically shift our weight so that we fall forward, catch ourselves with the free leg, and push off with our lagging toe, rolling our weight through to fall and catch ourselves again. All of this involves muscles and tendons and ligaments tightening and loosening at the right time, attached to a strong bony skeleton. There are some major downsides to moving this way and holding our body upright like this. For one, it puts an enormous amount of stress on our pelvic floor. You've got what is essentially an entire column of guts, of internal organs, being pulled down by gravity on a relatively small spot, which puts us at risk of prolapse more than most other animals. Walking on 2 legs is also less stable than walking on 4, making us prone to falls. And since our head is the highest point on our bodies because of our walking upright, it's got the longest distance to fall. And it also happens to carry some of the most important parts of us. Falling can and does kill us, and while having our arms freed up means that we can usually catch ourselves before we split our skulls open on a cave or kitchen floor, we don't always manage it. 684,000 of us die every year globally from unintended falls. We're also not very fast. Some of this has to do with the biochemistry of our muscles, but our bodies also just aren't really set up for the explosive high speeds that we tend to see in most other mammals. We make up for this, though, by being some of the best long-distance endurance walkers and runners in the entire animal kingdom. We regularly place for endurance walking and running, particularly in hot conditions, thanks to another ability tied to our way of moving, sweating. Humans have 10 times the number of sweat glands as compared to chimpanzees, meaning we stay cool like nothing else out there through evaporative cooling. The debuff of the sweat perk was recently found to be much slower wound healing time. Human wounds heal 2.5 to 3 times slower than those of our less sweaty relatives. There are countless physical benefits and detriments tied up with how we move, but I think the big question is, how and why and when did this type of system evolve that's so different from all other living primates? Humans and human-style bipedalism evolved from an ape which was not bipedal, but was probably already upright. During the Miocene, which began some 23 million years ago and ended some 5 million years ago, a ton of different ape species lived. I'm talking dozens of species all across what we consider the old world, so Africa, Asia, and Europe. And these guys were moving in very different and interesting ways, many of which involved hanging below the branches or walking on top of the branches on 2 feet in what's called suspensory clambering or extended limb clambering. This means that their foramen magnum was tucked forward in a way more similar to us than chimpanzees, because although they weren't bipeds walking around on the ground, their bodies were still held upright. 7 million years ago, the human or hominin lineage split off from our closest living relatives, the panins or chimps and bonobos, and sent us down a pathway to plodding. How do you turn an upright ape swinging around in the trees to a terrestrial biped? Well, you have to evolve enormous changes in the position of the foramen magnum, in the shape of the spine, in the morphology of the feet, and in the pelvic girdle. The pelvic girdle is one of the most striking differences between humans and most other primates, and the human setup is vital for how we move. It strengthens the pelvic floor and anchors large and powerful muscles necessary for our efficient bipedalism. In order to strengthen the pelvic floor and anchor those powerful muscles, like the glutes for example, we had to make huge changes to the form of the pelvis itself, particularly the blades of the pelvis or the ilia. The pelvis seems like a single bone, but it's actually many bones which are fused together or articulated at joints. The whole thing, the pelvis, is composed of the sacrum and the os coxae, which articulate with one another at joints in the posterior or rear and the anterior or forward parts of the structure. Each os coxa, singular of os coxae, is composed of 3 fused bones: the ilium, or blades of the hip, the ischium, the part that you sit on, and the pubis, the forward part with the cartilaginous joint. All of the pelvis is important, but for bipedalism, we need to focus on the ilium, or blades. This bone is critical for facilitating and stabilizing walking, and it looks very different in humans as opposed to other apes. In humans, these bones are short, wide, and run front to back in the sagittal plane, with the rest of the pelvic girdle creating a bowl shape. Pelvis literally means basin in Latin. This is very different from most other primates, even in more facultative bipeds, whose ilia runs side to side in the coronal plane of the body. Essentially, one of the first and most important steps in becoming a committed terrestrial biped is changing the ilium. You need to shorten it, you need to broaden it, and you need to rotate it into the sagittal plane. This seems like a tall order, evolutionarily speaking, doesn't it? Now, other animals have done similar remodels, just look at bats or whales, but it takes a special set of circumstances to pull off, especially because other dependent muscles, ligaments, tendons, nerves, blood vessels, and more must change in complementary but non-damaging ways. If humans evolved from more basal or primitive apes, we should see this transition occur in the fossil record. And we do. In spades. The lineages leading to humans and chimpanzees split around 7 million years ago, with the Miocene apes living before this split having pelvises that were pretty similar to many living apes, a tall ilium and ischium with the blades running side to side in the coronal plane. We can see this in the fossil pelvis of Ardipithecus, a Hungarian ape that lived about 10 million years ago. Fast forward 6 million years, just skip forward to Australopithecus, the genus that Lucy's species Australopithecus afarensis belongs to, and we have Competent, upright terrestrial bipeds already. Yeah, you know all those features that we talked about that humans have that allow us to be upright walkers when we were hanging around with my skeleton? Australopithecus, the genus, has the overwhelming majority of those features, meaning they were already committed bipeds. Now, this doesn't mean that they walked upright on the ground all the time. They still have some features that suggest they spent time in the trees. Long arms, curved fingers, and a more dorsally oriented scapula or shoulder blade. Essentially, this is because these guys had not invented fire yet, and they're not very big, so they probably used the trees to sleep in, like many apes do today, as well as perhaps to forage for foods like fruit. What I'm trying to say is they lived in an area where it was necessary to be competent and bipedal on the ground, but also to maintain some critical climbing abilities. From Australopithecus afarensis to Australopithecus sediba to Australopithecus africanus to Australopithecus prometheus, this genus was filled with competent upright walkers. From the first Australopithecus specimen discovered in the 1920s through the century all the way to now, Australopithecus has shown us that bipedalism evolved before big brains in our hominin relatives. And that's cool and fascinating and interesting, but it doesn't really answer our question today, does it? Because if Australopithecus was already a competent, upright biped, well, where are our transitional forms to that mode of locomotion in between the Miocene apes and Australopithecus? The answer would technically come in the 1990s, but it wouldn't arrive formally until the late aughts. Let's do a science exercise first. We know that of the pelvic bones, the ilium is perhaps the most important for obligate bipedalism. So which bone do we expect to change first? The ilium, the ischium, or the pubis? Probably the ilium, right? Ardipithecus is a genus of hominin that lived from 4.4 to 5.8 million years ago, the right age, and it's best represented by a marvelous partial skeleton which preserves the pelvis, reported on by White and colleagues in 2009. This specimen is incredible because it isn't just that it has a combination of arboreal and terrestrial or tree-dwelling and ground-dwelling characteristics, but some of the individual bones are themselves perfect transitions. The pelvis, shockingly, has a short, more human-like ilium, but a tall, more chimp-like ischium. This is combined with an anterior or forwardly placed foramen magnum, the hole at the base of the skull, that tells us that this animal was an upright ape like us, but is combined with a massively abducted chimp-like big toe for gripping the branches. It has smaller canine teeth than other Miocene apes, more like us, but long arms that would have made it right at home in the trees. This critter is a jack of all trades. Ardipithecus ramidus could move in the trees, or it could walk upright on the ground. So I'll ask you another scientific question. You can test yourself here. What kind of an environment would an animal like this live in? If you guessed one that was a mixture of tree patches separated out by swaths of grassland, you are correct. A mosaic species for a mosaic environment. Paleoanthropologists love that word. You couldn't ask for a more perfect link from the Miocene apes to the Australopiths. By Lucy and other members of her genus, we have short ilia and short ischia, an inline big toe, and more reasonable limb proportions, showing us that bipedalism was an early adaptation indeed. That's really cool, right? We see through the fossil record the change in the morphology of the pelvis, of the hips, in addition to all of the other changes necessary for bipedalism and big brains and all of the characteristics that make humans different from other living apes. And yet, scientists are never happy. It's great to know that something happened, and when it happened, and perhaps even why it happened, but that's not enough for scientists. They are gluttons for knowledge. We know that thunderstorms occur, and we know the conditions that they tend to form under, but what is actually happening when one is forming? How does humidity and pressure change to facilitate such an event? Transferred to biology, and specifically human evolution here, we know that the pelvis changed, and we may even know why it changed, but scientists want to know how that happened, step by genetic step. Today's paper is a genetic, developmental, and histological tour de force, and it's titled The Evolution of Hominin Bipedalism in 2 Steps. This was just published by Senavaratne and colleagues a month ago in Nature. It is an unbelievable paper, as you will see. We're going to go over some of it now because it's open access. As always, we begin with the abstract. Bipedalism is a human-defining trait. It's made possible by the familiar bowl-shaped pelvis whose short, wide iliac blades curve along the sides of the body to stabilize walking and support internal organs and a large-brained, broad-shouldered baby. The ilium changes compared with living primates are an evolutionary novelty. However, how this evolution came about remains unknown. Here, using a multifaceted, histological, comparative genomic, and functional genomic approach, we identified the developmental bases of the morphogenetic shifts in the human pelvis that made bipedalism possible. So they're like, what is actually happening when a human is in utero developmentally? What changes occur that send us down the human path for the human-shaped pelvis as compared to other primates or the mouse path, as we'll see in a second? They say, first, we observed that the human ilium cartilage growth plate underwent a heterotopic shift, residing perpendicular to the orientation present in other primate and mouse ilia. So they're like, all right, first what happens is the growth plate that is forming the pelvis shifts in a human as compared to other animals, right? So as an individual is developing in the womb, this shift happens by a change in the orientation and position of the growth plate that's going to become the pelvis. And they've actually gone and identified the specific genes, like where and when they turn on and how they're turned on to facilitate this kind of shift. They say, second, we observed heterochronic and heterotopic shifts in ossification that are unlike those in non-human primate ilia or in human long bones. So there's something unique about the ossification or how the cartilage turns into bone in the human pelvis as compared to the pelvis of other animals, or the pelvis, I should say, of other animals, as well as other spots in the human body. And I should say heterochronic and heterotopic just mean in an abnormal time and place. They say ossification initiates posteriorly, so in the rear, resides externally with fibroblast and perichondral cells, contributing to osteoblasts, and is delayed compared to other bones in humans and with primate ilia. So again, weird timing, weird place is how this ossification, or like the formation of bone, is occurring. Underlying these 2 shifts are regulatory changes in an integrated chondrocyte perichondral osteoblast pathway involving complex hierarchical interactions between SOX9, ZNF521, PTH1R, and RUNX2 to FOXP1P2. These innovations facilitated further growth of the human pelvis and the unique formation of the ilium among primates. Remember, Ardipithecus, our Miocene ape's pelvis, looks a lot like a chimp pelvis. So to go from the chimpy-looking pelvis to the Australopith/humany-looking pelvis, you have to make a lot of changes, and a lot of them surround the ilium. So this paper asked, what are those changes? Like, looking developmentally, what are those changes genetically? Which genes are turned on? When are they turned on? How are they turned on? And how does it differ in humans as compared to chimpanzees, to the mouse lemur, and to a mouse? So how they did this is pretty cool. They say here, using detailed histological, anatomical, and functional genomic approaches, we report evidence for the developmental genetic bases of the human ilium. Using this approach on human developmental samples and through comparisons with the pelvic development within a comprehensive evolutionary framework of primates, including chimpanzees, we reveal 2 major genetic innovations that shape the human ilium: a spatial heterotopic shift in the ilial cartilage growth plate orientation, and a dual timing, heterochronic and spatial heterotopic shift in the iliac ossification. Both innovations facilitated further pelvic growth and the unique formation of the human ilium among primates and possibly mammals. They talk about this more in the paper, but also in many of the articles covering this paper, but they used like well over 100 samples, like developmental samples that have been accumulated in various institutions in the United States and in Europe. And like some of these samples from various ages, right, of humans, chimps, the mouse lemur, etc., because these are very rare specimens to get a hold of, were like over 100 years old, like glass-mounted slides and stuff that's been sitting in formaldehyde jars forever. Like, it was a very labor-intensive project, I will say. So because they had samples for humans, chimps, mice, and mouse lemurs at various stages of development, so like at 1 week, at 2 weeks, at 3 weeks, at 4 weeks, and so on and so forth, they were able to track how the growth plate with regard to the pelvis changes at each major stage of development and how it differs in these organisms in question, in humans versus chimps versus the mouse lemur versus the mouse. And that was for like the major, like large scale morphologic changes, right? So like, what does the growth plate look like in all of these animals at 3 weeks versus 6 weeks versus 8 weeks? This is basically like developmental and macroscopic anatomical differences between these guys. But in addition to that, they say to examine this shift at the molecular level, we conducted single cell multiomics and spatial transcriptomics on microdissected ilia and adjacent soft tissues. And then they have some pictures of that. A whole lot of this methodology is completely over my head because this is so foreign to what I normally do, although I am very familiar with the comparative morphology here. That's what they did on the large scale. The comparative morphology, stage by stage, developmentally compared between humans, chimps, mouse lemurs, and mice. And they've got some really cool pictures here of these slices of various portions of the pelvis across these organisms, again, at different stages. And then they did this at the genetic level. So what are the large-scale changes and what are the genes that are causing these large-scale changes? Or how are these genes expressed which are causing these large-scale changes? And the reason for this, I think, is pretty intuitive. If you guys have had a biology course before, you'll probably remember that early in development, or rather the earlier you go in the development of an animal, the more similar any given animal you're going to look at is going to look to anything else, right? Like very early embryos are going to look the same across humans, mice, monkeys, a chimpanzee, a piglet, a cat, a chicken, a fish, right? And then as they develop along their respective paths, they will diverge from one another as their unique to the species genetic instructions run their course. So since the human pelvis differs from other primates, the question is, when does it start differing and how does that actually play out? How does the human pelvis begin to differentiate itself developmentally? The authors provide the genetic network for how the human pelvis differentiates itself, meaning it is changes in these genes and in their regulation that led to the human, the hominin, pelvis, and our unique mode of locomotion from an evolutionary standpoint. So what actually happened? To get a human pelvis from a chimpanzee pelvis, the ilium needs to shorten and then it needs to widen. And what the authors show in their work is that this didn't occur in 2 steps, first a shortening, then a widening, or first a widening, then a shortening, as may have been previously proposed, although I'm not sure of the history of that. Instead, the entire growth plate, which in a chimpanzee grows tall and thin, just rotates by 90 degrees. So simultaneously, you get a wider, shorter pelvis because of the direction that that growth plate is developing. I'm going to explain it with pencils in hopefully an accurate way. Imagine that this pencil is the chimpanzee, mouse lemur, and mouse growth plate, right? It's developing tall and thin. But in humans, this same growth plate rotates by 90 degrees. So it doesn't become short and then wide, it becomes simultaneously short and wide because of the rotation of that plate. They summarize it themselves in the paper saying this reveals that unlike the well-documented longitudinal development of human long bone growth plates, or of the pelvis in chimps, mice, and mouse lemurs, the ilium growth plate becomes aligned perpendicular to the initial bone axis, thus both widening the ilium and making it short in height. That's just wickedly cool, right? Two birds, one stone. Extended figure 1 shows graphically what they're talking about here. In a putative gene regulatory network underlying the dual structural novelties of the human ileum, we can see that in A here, we have our chimp, our mouse lemur, and our mouse, and we have our human here on the right. Here's the initial growth plate. It becomes tall and thin in these guys. It becomes short and wide in humans. And I'll add that this is the same figure that we got our gene regulatory network from. So this is really cool and epic, obviously, but it's not the whole story yet, because it isn't just that the human pelvis is short and wide, again, shown by this pencil in my hand here, whereas the chimpanzee pelvis would be tall and thin. The chimpanzee pelvis also has blades that run side to side in the coronal plane of the body, whereas in humans the blades run from front to back. As it turns out, the bowl shape of the human pelvis and that front to back sagittal orientation of the blades of the pelvis has to do with the timing of ossification. In human long bones, ossification starts in the middle of the bone and then extends bidirectionally. So you can imagine, again, this pencil, these are very useful today, is your femur or thigh bone. Ossification, bone formation begins in the center, right? So your little cartilaginous lattice is forming and then it starts to become bone first in the middle and then it extends in both directions, superiorly and inferiorly. The same is true for the pelvis, the growth plate of the ileum, in all of the primates and in the mouse looked at here, but such is not the case in the human pelvis, although it is how our long bones work and evidently how our pelvis used to ossify. But human ossification of the ilial growth plate looks very different. As you can see again in this figure, here is the growth plate for the chimp, the mouse lemur, and the mouse, and it is ossifying bidirectionally up and down. Whereas in the human, after we see the orientation of that growth plate change and it starts to develop as short and wide instead of tall and thin, the ossification starts like almost radially here. That's probably the wrong way of explaining it, but as you can see, it's quite different from what we're seeing in long bones or in the pelvic growth plates for these other guys. You're probably reasonably wondering, what does the ossification timing of the human ileum as compared to the ilia of other animals and the direction of that ossification have to do with going from a coronal side-to-side orientation to a front-to-back? Like, how does that actually happen? How does the timing do anything for us there? After again discussing the specific genetic pathways that are at play here for this development, in the discussion, they start to specifically focus on muscles. They say, nonetheless, we would be remiss not to consider the importance of the soft tissue environment in which the human ileum has formed. We found evidence that the iliac cartilage interacts directly with developing muscles that are especially important for bipedal walking, so your glutes, etc. We observed that the gluteus medius and minimus directly anchor via tendinous connections to cartilaginous ilia in the advancing AGZ. We similarly observed that both the rectus femoris and the iliofemoral ligament connect to the cartilage precursor of the anterior-inferior iliac spine before secondary ossification. Moreover, relevant muscle paramecium, and tendon cell types displayed some of the strongest signals for enrichment for evolutionary selection at these critical stages. Previous studies showed that during this morphogenetic window, iliac blades shift from being paracoronal to parasagittal. So it is during the window that they specifically looked at here when these muscle attachment sites and the nature of the ilium are interacting with one another and establishing themselves, that those ilia go from being coronal or side to side and they shift to being sagittal. That's critical. And here's the important takeaway here as I understand it. The adult configuration is that parasagittal configuration. Given the role of early prenatal muscle contractions on shaping skeletal development, and that RUN or RUN-X2 is known to be mechanosensitive, we posit that the early muscle anchoring and contractibility help to parasagittally orient the blade and potentially influence the pace of the anterior advancing parachondral ossification front via mechanotransduction. Here's how I understand it. I could be wrong, but this is my takeaway of that section. The human ileum at the stage that ossification begins has already taken that short, wide developmental milestone step, right? So it's not tall and thin anymore, it's short and wide, but it's still in the wrong plane. It's still running side to side. But because that ossification, which is, once a bone is ossified, it's much harder to change than when it's in its cartilaginous state. Before it's ossified, the muscle attachment sites are already beginning to establish themselves, and as those muscles develop, attach, and contract, which is evidently something that happens during this stage in development, they start to pull that blade, that ilium, into the sagittal plane. The muscles themselves, while it's still spongy-ish and cartilaginous, pull that blade into the human configuration, which it's able to do only because of that delay in ossification at the end portions of that ilium. So the human stages are get short and wide and delay ossification so that the muscles, which are still developing at their normal pace, can pull you into the proper direction and orientation. And that's how you get the bowl-shaped pelvis with the short, wide ilia. That's bipedalism in 2 steps. That is absolutely insane. That's so cool that they managed to map this out. This is like a functional pathway for one of the most important steps, in bipedalism. They did something very similar, and I made a video about it, for the loss of the tail. They're like, okay, we know when tails are lost, and we know probably why they're lost, and we see this happen in the fossil record, but how did that actually happen genetically? It's a good video, you should watch it. All right, let's wrap things up. They say during hominin evolution, bipedalism arose as a suite of morphologic changes, and they talk about the foramen magnum, the S-shaped spine, the valgus knee, the inline halix, etc. They say the fossil record informs us that not all of these traits arose simultaneously. We already went over that. Although fossil pelvies are not available for the earliest period of hominin evolution, 8 to 5 million years ago, it is noteworthy that some of the earliest bipedal traits are present in Ardipithecus and in Australopithecus, including a short and wide ilium. But how did this reconfiguration happen during evolution? One hypothesis is that the reorientation of the blade reflects a saltatory leap, underpinned by 1 or 2 molecular changes of large effect. So basically it was like one Ardipithecus was born and it had relatively major changes compared to its parent, like a large saltatory leap that gave that individual major advantages. They reproduced more often and passed that characteristic onto all of their kiddos. They say an alternative hypothesis is that the shift was gradual, mediated by the high polygenicity encoded in a complex pelvic developmental system. Regardless of mode, we envision a 3-step model. They say first, early on, so 8 to 5 million years ago, there was an abrupt or gradual shift away from the vertical to transverse growth by way of chondrocyte reorientation. This occurred as locomotion went from ape-like to facultative bipedalism, with muscle function shifting for the need for increased lateral stabilization. So this is going to be your immediate shift of going from tall and thin ilium to short and wide ilium. And this is going to happen pre-Ardipithecus, because we know Ardipithecus already has the right ilium, but the ischium and several other characteristics are still very ape-like. This makes sense. Ardipithecus ramidus is a facultative biped. I should add here that they're using a stricter definition of facultative bipedalism. They're talking about it more as something like Ardipithecus, something that is spending, more than 10% upright on the ground, walking bipedally. It's annoying that people can define things in different ways, even in science, but we work with what I have. I think as long as you're clear with what the terms mean when you're using them, it's okay. Second, later, 5 to 2 million years ago, as hominins shifted from facultative to obligatory bipedalism, this growth plate orientation became fixed or locked in by additional or new molecular changes, leaving a remnant signal of vertical differentiation. So this is pretty important, right? Because we still see that original vertical differentiation really, really early on in human development. It's like how we have a tail for a little bit, and we have our little pharyngeal gill slits that look like gill slits for a little bit before they differentiate again along the human pathway of development. I'm being a bit crude there, but hopefully you get what I mean. During this stage, we predict a concomitant ossification shift to the posterior ilium to permit anterior growth in response to muscle function. Third, more recently, 2 million years and onward, with the increasing demand for bipedalism for running, and as fetal heads and shoulders became larger, the timing of ossification became delayed to enable enhanced growth and the retainment of a complex iliac shape. According to this model, we argue that the ilium is part of highly important adaptive musculoskeletal pelvic complex that is shaped during evolution under the constraints of complex polygenic underpinnings. I think that sums things up beautifully, probably more concisely than I did over the course of this video. But the human pelvic shape is facilitated by a change in the orientation of the growth plate of the ilium, followed by changes in the directionality and timing of ossification that allow that growth plate to sort of pull forward and give it that nice bowl shape that allows us to move upright efficiently and still give birth to our kids. And this is supported by not just macroscopic changes observed in the developmental differences of humans versus other closely related animals, but down to the genes which are responsible both in their presence and in their regulation for these changes. This is demonstrated. That's absolutely insane. And better yet, this pathway, this genetic pathway and the developmental differences that we see looking at the gross morphology differences reflect the changes that we see in the fossil record. Wow, corroboration. Who would have thought? So now we've got the developmental and genetic basis for the loss of the tail in apes generally and the weird pelvis of humans, at least with regard to the ilium. And I suspect that we are going to see this exact type of study be carried out for all the unique human characteristics or unique ape characteristics. Eventually, we're going to have a step-by-step genetic pathway, genetic model for how you get a human from a generic Miocene ape, and probably for that matter, how you get a chimp and how you get a gorilla and how you get an orangutan. I can't wait. It's gonna be awesome and very, very interesting. If you like this video, and if you like what I do generally, please consider supporting the channel in the free way. That's liking, commenting, and subscribing, and ringing the bell. Asking makes people do it. And if you want to support the channel in a more financial way, you can join my Patreon. Patrons get early access to videos when I'm on top of things. But really, I just want to thank you for being here, for being such a curious primate. Obviously, I'm thanking you from the bottom of my gentle and, of course, very modern heart. And folks, I'll see you real soon next time.